The first session will be co-chaired by Professor Hidenori Arai, who is the president of National Center for Geriatrics and Gerontology in Japan, and by Professor Lin Qihong, who is the president of National Yangming Jiaotong University.
Okay, so good afternoon. My pleasure to moderating with my old friend, Professor Arai, for the session. The first speaker this afternoon is by Professor Tsai, Tsai Tingfen, and basically says Okay, so Professor Tsai is currently the distinguished professor in previously National Yangming University and right now the National Yangming Jiaotong University. She's world-renowned. I don't know you want to call yourself gerontologist or a cell biologist still, okay? And she's gonna give us a very interesting talk about rejuvenations, turning back time by enhancing 62 prolongivity gene. So let's welcome Professor Tsai.
Thank you for the introduction from President Lin. Today I'm going to talk about introduce the E2 prolongivity gene and update the progress in these longevity genes, and for all the friends, including the friends from Japan online and Toba. The CDGSH ion sulfur domain are in short CISD protein families. And currently there are three families, there are three genes in the families, including the 61, 62, and 63. 61 is located in the mitochondrial outer membrane. However, 62, it has a very similar structure with the 61, have transmembrane domain, and the ion sulfur binding CDJS ion sulfur binding domain is located in the mitochondrial outer membrane, the ER, and the mitochondrial associated ER membranes. For the human 62 homodimers, each monomers that contain 2 ion and 2 sulfur clusters. And this ion sulfur cluster traditionally is responsible to accept and release the electrons. Based on the human aging genomic resources, and currently there are 8 prolongivity genes in the mammals. And the 62 is one of the prolongivity genes in mammals, and this actually is based on majority the study published from my lab. Regarding the function of 62, 62 actually regulates the calcium homeostasis and the mitochondrial functions. And for example, in the hepatocytes, 62 will interact with the SERCA2B, and SERCA is a calcium channels that will pump the calcium from the cytosolic into the ERs, so to maintain the calcium homeostasis. And the 62 also located in the mitochondrial outer membranes, and to maintain the mitochondrial function. So during haploinsufficient 62, and this will lead to increase of the oxidative modification and decrease of the calcium channel activity of the SERCA. So this will cause the increase of cytosolic calciums and leading to the mitochondrial calcium overload and the dysfunction. And this increased cytosolic calcium also will turn on the ER stress and lead to fatty liver development in the liver. So 62 deficiency will cause a variety of age-related disease. For example, that will cause the corneal degenerations, sarcopenias, heart failures, arrhythmias, as well as the fatty liver development just mentioned, and the adipose tissue atrophy. In addition, the 62 deficiency will accelerate the A-beta mediated neuronal loss. And interesting, during the natural aging of wild-type mice, the 62 expression level is down-regulated. For example, in the muscles, at the middle age, at 13 months old, the 62 decreased to about 30% compared to the young mice at 3 months. And at the very old age, about 26 months old, the expression level is less than 20% in the old age. compared to the young age. And similar observation was observed, for example, in the heart and other organs. So indeed, the 62 knockout mice is an accelerating aging mice. In contrast, a persistent high level of 62 expression in the 62 transgenic mice, actually this mice is a very long-lived mouse. For example, look at the right mouse. This 62 transgenic mouse is at 34 months old. This equivalent in the human is about 105 years old. It looks younger compared to the wild-type mice at 15 months old. So this is the survival curve for the mice. So the blue line is the 62 knockout mice. The 62 expression level is 0. And the gray line is the heterologous mice. This is 50% of the 62. And the open box is the wild-type mice. It's 100% of 62 expression. The green line is the transgenic mice. They have 200% of 62 level. So based on this survival curve, obviously we have demonstrated there is a 62 dose-dependent increase of survival rate. So in the mice, the level of 62 decreased during natural aging. If we can maintain a consistent high level of 62 expression, then we can extend a healthy lifespan. So our purpose is to develop an anti-aging drug using the 62 as a molecular target. So we have generated 62 reporters to monitor the transcriptional activation of 62. We use a human 62 bacterial artificial chromosome. This bag contains more than 100 kilobase of the DNA that contain the entire 62 genes, as well as the upstream and the downstream sequence. So we knock in the luciferase, this reporter gene, into the exon 2 of the gene to establish the 62 reporter. So this is the 62 reporter mice, okay? And under specific treatment, the increase of the luciferase actually indicate the activation of the 62 gene. And this specific treatment, actually, I tell you the secret, is the starvation. So starvation will activate the 62 longevity genes. And we also established two 62 reporter cell platforms. The first one is the mouse embryonic fibroblasts established from the transgenic mice. The second one is the HEC293 that contains the 62 back reporter cells. So regarding the compound libraries, we established a compound libraries from the noble herb that described in the classic of herbal medicine in Chinese, 神农本草经. And actually, this is a small compound libraries that's established only from the 60 noble herbs. And this compound libraries contain only 780 samples. But actually, there is a lot of the 62 activation inside. So this is a candidate list of 62 activators. For each of the 62 activators, we give a code name called PenZu, in short, PZ. So who is the PenZu? PenZu actually is a very long-lived celestial beings in Chinese myths. So that indicate they have a very healthy aging and longevity. So PZ19 is the compound I'm going to talk about. So PZ19 is a pure compound. The purity is more than 95%. And it is a very potent 62 activator that can activate or stimulate 62 expression in both the 62 reporter cell and the reporter transgenic mice. So we provide the PZ, these compounds in the food to treat the mouse for a long time. And in the mouse, the dose we use is 100 milligram per kilogram per day. This equivalent in the humans is about 490 milligram per 60 kilogram per day. So definitely this is a dose that achievable in the human. This is the protocol for the late life treatment of PZ19 in the naturally aged mice. We treat a very old mice, okay, at the age of about 21 to 22 months old. So this equivalent in the humans is about more than 17 years old. We treat the mouse for 6 months and obtain the sacrifice the mouse and obtain the tissues at around 27, 28 months old. So this equivalent in the human is more than 80 years old. So interesting, we got very exciting result. For panel B, this is the Western blot analysis for the level of 62. So we after quantification, you see, for example, in the skeletal muscles, the 62 level downregulate to about 30% at old age. However, PC19 treatment for 6 months, they can significantly increase the 62, the longevity genes, to a level that is comparable to the young mice. And similar observation was observed in other organs, for example, in the heart. And most strikingly, we found they increased the median lifespan, and in particular, this is a healthy lifespan. So the blue lines is the mouse treated with the PC, started at 21 months old. And the dark lines is the vehicle control. Gray line is untreated. So this data demonstrate that PC treatment can increase the median lifespan for about 9%. So this equivalent in the humans in Taiwan, for examples, the male will increase the median lifespan from 78 years old, plus 9% is increased to 85 years old. In the humans, is increased from 84 years old to more than 90 years old. So I think this is quite impressive, at least to me. And this is monitor the whole body energy metabolism. So this data here is, we monitor the daytime and the nighttime. And for the mouse, they are active during the night times. And the green line is the control from the young mice at 3 months old. Blue line is the old mice. Okay, so, and the red one is old mice treated with the PC19. So this data actually demonstrate that the PC19 can increase the whole body energy metabolism in terms of oxygen consumption, and similar observation for the CO2 production and energy expenditures. And most strikingly, we monitor the same group of mice, okay, when they are different at different age. So again, the green line is the young mice control, and the mouse, the orange line is the mouse at 23 months old. Dark line is at 28 months. So this data indicate there is an age-dependent decrease of the whole body energy metabolism. However, when we treat the PC19, you look at the dark line, this one is at 28 months old. The light blue is 23 months old. So this data actually indicate the mouse is getting older, but the whole body energy metabolism is getting better. So this data actually is demonstrated that PC19 appear to rejuvenate the aged mice in terms of whole body energy metabolism. We also apply the micro-CT to monitor the whole body fat and body limb. So actually these data demonstrate that the PC19 treatment that can reduce the fat accumulation. At the same time, they can increase the body limb compared to the vehicle control. And this is monitor the glucose homeostasis. And regarding the body weight, there is no difference for the body weight with or without PC19 treatment. And interesting, for the blood glucose, after the fastings, the blood glucose level is significantly reduced after the PC19 treatment. So actually, during the natural aging, there is a panel of differentially expressed genes associated with glucose metabolism. They are actually upregulated during the natural aging. So this problems explain why the basal blood glucose is upregulated during aging. However, after PC19 treatments, these differential expression, they are downregulated. So this is a summary to summarize that the PC19 actually slow down or even rejuvenate the heart ages in the naturally aged mouse. So if the young mice in the heart, the 62 level is 100%. When they're getting old, at 24 months old, the level of 62 downregulate to less than 50%. And for this transgenic 62 transgenic mouse, this is a long-lived mouse models. And the expression level at the old age is 100 to 30%. Very interesting, the old mouse treated with PC19s, the 62 levels that can upregulate to about 130%. So this level is similar to the long-lived transgenic mice. Okay, so for the monitor of cardiac functions, we found that the 62 activated PC19 that indeed can improve the aging-related dysfunction. Regarding the structure, the PC19 can improve the structural damage and the mitochondrial functions. And this is a summary for the skeletal muscle aging, and very similar, we found the PC19 can slow down the skeletal muscle aging. So this is the RNA sequence analysis. And it looks like quite busy, but we can just look at the colors. For panel A, the left one is blue one is the panel of differential express gene expressed in the young mice. The second is the line is the old red one. That means in the old age, in the heart, they are upregulated. However, these old mice, after PC19 treatment for 6 months, the differential express genes are downregulated. Okay, and we observe this in the heart and similar observation in the skeletal muscle. So this RNA sequence data actually demonstrate the PC19 that can indeed bring about a younger transcriptome patterns of gene expression during the old age. So one important question is that does PC19 delay the agents that is function in a 62-dependent manners? So to answer this question, we knock out the 62 genes in the muscles. We look at this blue box, okay? In the absence of 62, actually when we treat the PC19 totally, the beneficial effect is disappears. So this data actually indicate the PC19 delay the skeletal muscle aging indeed is in a 62 dependence. So this is another evidence from the RNA sequence. From the RNA sequence data, we know that about 80% of the differential express gene, the pattern will disappear once the 62 is absent. However, there are about 20% of the differential express gene that maintain their patterns even in the absence of 62. So that means 20% of the differential express gene, they are 62 independent. But majority of differential express gene, they are dependent on the presence of 62. So the first part regarding the healthy aging and longevity, the conclusion is the PC19 that can promote the longevity and delay aging via activation of the 62 in the naturally aged mice. So the second part I'm going to talk about is the 62 activator and the skin agents. And this part is still unpublished data that they are manuscript under the revision. For the humans epidermis, most of the cell, 90% of the cell, actually they are keratinocyte, okay? And regarding the epidermis layers, okay, from the buttons, these are the basal layers, and then spinous layer, granular layers, and the conified layers. The basal layers contain most of the keratinocyte. They are proliferating keratinocyte. And they have a marker K14. This is for the basal layer. And this is the immunofluorescence staining of the 62 and the K14s using the human skin samples. So this data clearly demonstrates that the 62 actually is mainly expressed in the proliferating keratinocyte of epidermal basal layer in the human skins. If we obtain the skin tissue from the sun-exposed and the non-sun-exposed regions, the sun-exposed region, for example, the face and the neck, the non-sun-exposed region, for example, from the chest, okay? And this immunostaining showing very interesting data is the 62 actually they are downregulated in the sun-exposed sites compared with the non-exposed sites. The 62 often downregulate in the sun-exposed tissue of the skin. So one important question here is the PC19, they can improve UVB-induced skin photoaging. And if this is function in a 62-dependent manner. So we use the wild-type mice and the knockout mice at 3 months old. We pre-treat the mice for PC19 for 7 days. After 7 days, we challenge the mouse with UVB irradiation once a day for a sequential 5 days. And after 2 days later, we sacrifice the mouse and examine their skins. So interesting, panel B is a Western blot analysis. Obviously, you can see the UVB irradiation will downregulate the 62 levels, and to about the quantification shows that downregulate to about 50%. However, with the presence of the PC19, the 62 level obviously is upregulated to a level actually there is seems like is stronger than the untreated the skins. And the other marker is MMP1. MMP1 actually is a marker's major indicator for the UVB-induced damage in the keratinocyte. So this data clearly demonstrate that PC19 treatments that can significantly reduce the MMP1 that indicate that reduce the damage in the keratinocyte. So this is the dorsal skin from the mouse. Okay, after UV irradiations, what we see is the skin has a redness and they have a thick wrinkles. However, with the PC19 treatments, look at this skin. They look smooth and unwrinkled. Interesting, in the 62 knockout mice, this protection to the skins disappears. And because after PC19 treatment in the 62 knockout mice, the skin still look reddish. So this data clearly demonstrates that the PC19 indeed can ameliorate the UVB-induced skin damage and function in a 62-dependent manners. This is the histology staining with the Mason trichomes. So the mouse skin without any treatments, this is the mouse, the histology. After UVB irradiations, you see the skin, we see an increase of skin thickness. And this increase of skin thickness including the epidermis and the dermis. However, with the PC19 treatments, you see the thickness of skin is significantly decreased. So the next question is if that can rejuvenate or delay the skin aging. So this again is the same protocol we apply to see the healthy longevities. And this time we look at the skin ages. So interesting, similar to the skeletal muscle and heart. Here what we see is the level of 62 decrease during the aging, that decreased to about 50% at the middle age and decreased to less than 30% at old age. And interesting, in the old mice, treated for PC19 for 6 months, that can significantly increase the 62 in the skins. And this increase to a levels that is similar, comparable to the young mice without any treatment. So this is a very amazing data. So we look at the histology of the skin. Panel at this one. This is the skin histology from a very young mice at 3 months old. You see a lot of hair follicle on the inside. Probably you cannot see, but there is the spatial gland. Spatial gland is the lipid-producing structures. And then the bottom one is the old mice, the skin from old mice. Obviously, you can see the degenerate hair follicle and degenerate of the spatial glands. However, after 6 months treatments, this is the skin. What did you see? What you see is the increase of the hair follicles. And also sometimes we see a hair follicles associated with 2 spatial glands. So this data is obviously demonstrate the PC19 appear to rejuvenate the naturally aged skins in the old mice. We quantitate including the hair follicle density, the nucleus of the spatial gland, or the size of spatial glands. All of this data actually demonstrate that PZ19 not only slows down the skin aging, but also rejuvenate the naturally aged skin. So I'm going to tell you the secrets. What is the PZ19? PZ19 has two structural analogs. One is called PZ19A, and one is called PZ19B. PZ19A equivalent to hesperidin D, okay? The other one is hesperidin, is PZ19B. And the PZ19A hesperidin is present in the peel of the citrus fruit. So I think whenever you eat the oranges, usually you throw away the peel. And the secret for longevity actually is in the peel. So remember next time, don't throw away the peel of the orange, okay? So, okay, hesperidin is we call PZ19A. This structure has two sugars. And because of these two sugar residues, so it is not easy to be absorbed by the intestine. However, PZ19A, the hesperidin, can be converted to hesperidin, the PZ19B. And so move out, the sugar will be deleted, removed. And this PZ19B, hesperidin, actually is the very potent SIS-D2 activators. So this is what just mentions, all of the beneficial effect is come from the SIS-D2 activator, hesperidin, okay? And some of the probiotics include the lactobacillus, or include and also the bifidobacteria. These two genus of the bacteria, some of them that can convert this hesperidin to hesperidin. And actually, we have two INCI names approved international. Okay, one INCI name is for hesperidin, and one is for hesperidin. So what is the INCI? That is a requirement when we develop the anti-aging cosmetic. INCI is the international nomenclature of cosmetic ingredients. So we have these two INCI names, and we have technology transfer to biotech companies. And the biotech company actually have developed the anti-aging cosmetics. So you probably can search from the Googles. The name is AG Lab, okay? This means anti-aging, okay? Oh, I would like to acknowledge my SIS-D2 team members, including the collaborators for the translational medicine and drug development. Okay, I think I'm going to stop here, and I wish everybody have a healthy aging and a longevity. Thank you.
So thank you very much, and we have about 2 minutes left. May I ask a question? Do you have a tropical use formula for this your ingredient, your active compound, for
Yes.
topical use instead of
The anti-aging cosmetic actually is for topical use.
Right, but all your mouse model is systemic.
Yeah,
Oral.
that's a systemic. And actually, I didn't show the data. Systemic is for the whole body, is health. Okay, for the topical treatment, only for the skin.
Okay. Another thing is that you have in your liver seems that there is fatty accumulation, or you want to call it fatty liver. I just wonder, is this cause a problem for systemic use? Is there any hepatic impairment, functional impairment?
Yes, the functional impairment in the liver is during its age. Is caused by aging. Okay, and because when we deliver the PZ19 from the food, and then the first time, so after absorption from the intestine and then the circulation to the livers. So the first line beneficial, what we see is in the livers. So after like a few days, 3 days treatment for the PZ, we see the improvement in the fatty livers.
All right, thank you.
Okay.
So
I think hesperidin is already in the market in Japan, so the hesperidin is available. So I think the company is very happy to see the data.
But one thing I want to remind is hesperidin is not a strong SIS-D2 activator. They need to convert into hesperidin. So you need to have a good bacteria in your intestine.